Dear students, welcome to BioRatna. My name is Chandana Sengupta and our today's topic of discussion is the packaging of DNA helix. The packaging of DNA helix. Now the question is, why do we need to package the DNA? Why do we need? We means the cell needs. Why do we need to package the DNA? In case of prokaryotes also, in case of eukaryotes also. What is the problem? Let us see. DNA is a polymer of nucleotides. DNA is a polymer of nucleotides and it has two strands. And let us draw some nucleotides. This is one sugar, another sugar, another sugar. Three sugars we have drawn. Each sugar is attached to its phosphate and its nitrogen base. This makes one nucleotide. This is another nucleotide, this sugar, its phosphate and its nitrogen base. Now come to the third sugar. This sugar, here it will be attached to its phosphate group and nitrogen base and so on. Okay, let us draw this phosphate too. And this continues. Now, there is another strand. This strand, it will also have sugar, means nucleotides. This sugar, it is having its nitrogen base and its phosphate group. One nucleotide. Similarly, with the other sugars we will do, and then we make the hydrogen bonds here. If it is A and T, then double bond. If it is C and G, then triple bond. Now see here, this is one nitrogen base pair. We are going to use the word base pair. Where to use the word base pair? Where to use the word base? This should be clear, na? This diagram will help you in that. These are two base pairs. This is one base pair and this is its consecutive next base pair. And the distance between these two, the distance between the two consecutive base pairs, this is 0.34 nanometer, or we can convert it into 0.34 into 10 to the power minus 9, and we have to write the unit too, so meter. We are actually calculating the length of the DNA. When you know the distance between the two consecutive nitrogen base pairs, then it will be easy for you to calculate it when you get the number of base pairs. What is the total number of base pairs? Fine. जब total number of base pairs आपको मिल जाएंगे and you multiply it with this distance, then you get the length of DNA. Total number of base pairs in a typical mammalian cell, is how much? 6.6 into 10 to the power 9. Here we are writing the number of base pairs, so we will use base pair. So we have written 6.6 into 10 to the power 9 base pairs. When we multiply these two, then this comes approximately 2.2 meter. 2.2 meter. Thus the length of the DNA comes to 2.2 meters. And what is the dimension of nucleus? It has to fit into the nucleus, na? The dimension of nucleus is 10 to the power minus 6 meter. Too small. That is the reason why we need to do the packaging of DNA. First we will see it for prokaryotes and then we will see for eukaryotes. First we see the packaging in case of prokaryotes. You know that a prokaryote cell, it does not have a nucleus. This is a prokaryote cell with its plasma membrane and the cell wall. Then though there is no nucleus, the DNA is not scattered in the cell. DNA has negative charge. You remember why DNA has negative charge? It is because of the phosphate group present in the DNA. Now that means it will be attracted to positive charge. So DNA is negatively charged and it is held at a place by positively charged proteins in the form of large loops. These are the large loops in a place called nucleoid. It does not have nucleus, but the DNA is not spread out. It is held in place in the form of these large loops. Let us make it double-stranded. Okay, this is it. So it is held in place by positively charged protein molecules. This is in case of prokaryotes. Now let us move to eukaryotes, which is a bit more complicated. Now we study the packaging of DNA in eukaryotes. In case of eukaryotes, this process is a bit complex. DNA is negatively charged molecule, so the principle remains the same. That is, now the DNA will be held together by positively charged proteins called histone proteins. These are histone proteins. First, we will see why do histones have positive charge. Why histones have positive charge? What is the reason of histones being positive? After that, we will see the formation of histone octamer and then the formation of nucleosome. So first we see why histones are positively charged. Positively charged. You know the reason of DNA being negatively charged? The negative charge is present in the phosphate group of DNA. Here, histone is a protein. Protein is a polymer of amino acids. So naturally, the protein will become positive or negative according to those amino acids which are in abundance in that histone protein, which have the positive charge in their side chain. In simple words, the protein will become positively charged when it is having the amino acids which have positive charge in their side chain. So these two amino acids which have positive charge in their side chain, which are present in abundance in histone proteins, they are arginine and lysine. This arginine and lysine, they have positive charge in their side chain and they are present in abundance in the histone proteins and they provide the much wanted positive charge to the histone proteins. Now let us see how histone octamer is formed. The histone octamer. The structure around which DNA wraps, it is a round ball-like structure. We call it bead-like structure. So this is not a single unit. It is made up of 8 units. That's why it is called octamer. It is having 8 units. That's why it is called octamer. And what are these 8 units? 2 units each. 2 units each of what? H2A, H2B, H3 and H4. So 2 of each, that makes it 8. These 8, they come together and they form this Rubik's cube, that is the histone octamer. So this is histone octamer. Understood the formation of histone octamer? Now we have to see how from this when the DNA wraps around it, the nucleosome is formed. Only this bead-like structure is histone octamer. Now the DNA molecule, it will wrap around this nucleosome. Okay? And here there is one safety pin. Na? Here there is one pin which will hold these threads in place. And this DNA, what amount of DNA has wrapped around the histone octamer? It is 200 base pair. So this is 200 base pair of DNA and this histone octamer together. This is now called the nucleosome. Thus, here now we have studied why the histones have positive charge. Then first we have seen the formation of histone octamer and then we have seen the formation of nucleosome. This nucleosome is the repeating unit in the packaging of DNA. Now further we will see the bead and string arrangement and how further packaging, this is not the final packaging, how the further packaging of DNA is done. Now let us see what is beads on string arrangement. Beads on string. You have already studied about the bead. That bead is having one histone octamer and 200 base pairs of DNA. That is, this is the histone octamer. I am again drawing the diagram because about H1 I have not told you. This is histone octamer. This is the DNA strand wrapped around it, and the pin we were talking about, which is holding the two threads of DNA. This is H1. This means this histone octamer, it has H2A, H2B, H3 and H4. Remember all these points, students. These are important for MCQs. And the H1 is outside. And this is the DNA strand having 200 base pairs. Fine. So when we observe the chromatin, what is chromatin? Chromatin is the diffuse state of the genetic material. Diffuse state of genetic material, when the cell is not dividing, then at that phase, when you see the genetic material, it is in a diffuse state called chromatin. जब इसे आप देखते हो, then you see these nucleosomes. These are the nucleosomes, and this string is only DNA. This is linking these two nucleosomes, isn't it? It is linking the two nucleosomes. That's why it is called linker DNA. This is beads on string arrangement, having all the nucleosomes and the linker DNA in between. This linker DNA, how many base pairs it has? It has 60 base pairs. The linker DNA, it has 60 base pairs. Done? This is not the final packaging. After that, this bead on string structure, it undergoes further compaction. it undergoes further compaction to form the chromatin fiber, and this chromatin fiber, it will undergo further compaction and it will form the chromosome. This chromosome, at which stage it is most compact? You remember cell division? At which stage the chromatins are most compact and the chromosomes are most thickest and smallest, that is metaphase. So during metaphase they are most compact. This is how the final compaction is done and we get the chromosomes. Now, if you think that this is again because of histone proteins, that is not true. Let us see who is responsible for this further compaction. Now we will see who is helping for the further compaction. That is NHC protein. What is NHC protein? Histone you have studied. This is non-histone. This is non-histone chromosomal protein. So this is non-histone chromosomal proteins which help in the further compaction of the chromosome. This is it. Then when the final packaging is done, when you observe the chromosomes, then at some places you will find that it is very compactly packed. It is very compactly packed, whereas at some places it is not that compactly packed. This part, it is called heterochromatin. It is very compactly packed. The two strands do not open up and so they do not take part in transcription. When they are not taking part in transcription, so naturally they are not going to form mRNA, they are not going to form the proteins. So this is transcriptionally inactive, whereas this part which is not that compactly packed, this is called euchromatin and it is transcriptionally active. This is not active transcriptionally and this is active transcriptionally. So if you think that heterochromatin is not doing any work, is not of any importance as it is transcriptionally inactive, then you are in for surprise and we will see its many applications, one in DNA fingerprinting. So I hope this packaging of DNA helix was clear to you. If it is so, and if you are new to my channel, then do subscribe, like and hit the bell icon so that you get all the notifications in time. Thank you for watching the video.